I welcome you all to today's session. In this session, we'll be discussing about Chapter 5, Fundamental Unit of Life. And this chapter deals with cells. Cells, as we know, they are the structural and functional unit of life. All tissues in our body are composed of these units called cells. Tissues combine to form organs, and these similar organs will combine to form an organ system that make up the entire body. So in a way, we can say that cells are the units of life. They are the building blocks of life. Everything that is present in our body is either made up of cells or requires cells for its functioning, right? So it is the structural and functional unit of life. Now, if we start with the discovery of cell, when was cell discovered for the first time? In 1665, Robert Hooke, he observed the cells of a cork, which is the, a dead cell, through a self-designed microscope. And he observed that the structure of the cells, it resembled a honeycomb, right? And it consists of many little compartments. Many little compartments were observed, and these compartments or these boxes, they were termed as cells. So for the first time, a dead cell, it was not a living cell that was observed, a dead cell was observed by Robert Hooke in 1665. And he observed that there are compartments that make up the entire cork, and these compartments were termed as cell. In 1674, Leeuwenhoek for the first time discovered living cell, right? For the first time, he discovered living cell in the, there were certain organisms present in the pond water from which he discovered living cells. Nucleus, which is one of the most important structures which is present inside the cell, was discovered by Robert Brown in the year 1831. Purkinje in the year 1839, he coined the term protoplasm to represent the fluidic or the watery substance that is present inside the cell. The cell theory was given by two scientists, two biologists, Sleeden and Swan, in the year 1838 and 1839. And the cell theory says that all the plants and animals, in fact, all living organisms that are present, they are composed of cell and the components of cell. And cell is the basic fundamental unit of life. It is the structural and functional unit of life. All the structures inside our body are made up of cell. This cell theory was further expanded by Virchow in the year 1855, and he said that all the cells, how do new cells come into picture? How are new cells formed in our body? So to that, he explained that all cells arise from pre-existing cells. Already existing cells, they divide and they give rise to new cells. And that is how new cells are formed inside our body. Now, based on how many cells the organism is comprised of, the organisms can be divided into two types: unicellular organisms and multicellular organisms. So unicellular organisms, they are comprised or they are composed of one single cell that is their entire body, and that single cell is involved in performing all the functions of that organism, from respiration to digestion to excretion. That single cell is responsible for performing all the functions, right? So amoeba, chlamydomonas, bacteria, paramecium, all these are composed of one single cell, and these organisms that are made up of one single cell, they are known as unicellular organisms. When we talk about multicellular organisms, as the name itself suggests, are made up of more than one cell. So many cells, they group together and they form a multicellular organism. And in multicellular organisms, because more than one cell is present, cells perform specific functions. So groups of cells that are similar with each other, they perform one specific function, right? So there is a distribution of labor in case of multicellular organisms that we observe, right? Plants, animals, fungus, all of them come under multicellular organisms. Now, there are different types of cells that are present. There are different shapes, different sizes of cells based on the function that they perform, right? So cells can be of different shapes and different sizes. There is not one standard shape or size for a cell. And each cell, it has certain components present inside it. It has certain specific components present inside it that is known as cell organelles, right? So these cell organelles that are present inside the cell, they are associated with performing a specific function. So numerous cell organelles are present, for example, nucleus, mitochondria, there is Golgi apparatus, there is endoplasmic reticulum, vacuoles, plastids in case of plants, etc. And these organelles, they are associated with a specific function that they perform. And these organelles together constitute the basic unit of life that we call cell. Firstly, we are going to talk about the plasma membrane. Plasma membrane is also known as the cell membrane. It forms the outermost covering of the cell. It forms the outermost covering of the cell in case of animal cells, and in case of plant cells, the outermost covering is cell wall, but after that, the cell membrane or plasma membrane is present. Cell membrane acts like a separation. It separates the content of the cell from its external environment. So, for example, this is the cell. This is the plasma membrane of the cell. This represents inside the cell, and this represents outside the cell. So, plasma membrane sort of separates or forms a demarcation between the internal environment of the cell and the external environment of the cell. And plasma membrane is a selectively permeable membrane. Why do we say that it is a selectively permeable membrane? Because it will allow the entry and exit of only some specific molecules in and out of the cell. It does not allow the entry or exit of all the molecules, right? It selects, and there are only certain molecules that can enter into the cell. There are certain molecules that can exit out from the cell. So cell membrane is a selectively permeable membrane. It will allow the entry and exit of only some specific materials, molecules, substances in and out of the cell, right? Now, there is a process that we need to study about, which is diffusion. What do we mean by diffusion? Diffusion is the movement of molecules from a region of its high concentration to a region of its low concentration. For example, I have a region A and a region B. In region A, let's say there are 50 molecules of carbon dioxide present. In region B, there are around 10 molecules of carbon dioxide present. So region A represents high concentration of carbon dioxide. Region B represents low concentration of carbon dioxide. So carbon dioxide will automatically on its own move from its area of high concentration, which is region A, to its area of low concentration, which is region B, till an equilibrium, a net equilibrium is achieved, and this is known as diffusion, right? So the movement of substances from its region of high concentration to its region of low concentration across the cell membrane is known as diffusion. It does not require any energy because the movement is happening along the concentration gradient. It is happening from high concentration to low concentration. And diffusion is actually one of the main reasons how gaseous exchanges take place in between the cells and in between cell and its external environment. Most of the gaseous exchange that we see takes place because of diffusion, right? Next, water also obeys this law of diffusion, which means water will also travel from its area of high concentration, where water is present in more concentration, more quantity, to the area of low concentration. But this special type of diffusion is known as osmosis. So osmosis is defined as the movement of water from its area of high concentration to area of low concentration through a selectively permeable membrane, right? Through a selectively permeable membrane. So water would always try to move from its high concentration to low concentration. Now, there are three types of situations and solutions that we can see. For example, there is a situation in which a cell is present in a solution, and outside the cell, the concentration of water is high. Water is present in high concentration, and inside, water is present in low concentration. There is less amount of water inside the cell as compared to outside the cell. So what is going to happen in this case? Water is going to move inside the cell. So from the outer environment, water is going to move inside the cell, and the cell is going to swell up in this case. And this type of a solution in which the water concentration is high is known as a hypotonic solution. And if we put a cell in a hypotonic solution, it is going to swell up. If we reverse the situation, outside the cell, the water concentration is low, and inside the cell, the water concentration is high, water is actually going to come out of the cell. There is going to be a net movement of water, but more water is going to come out of the cell, because inside the cell, water concentration is more, outside it is less. So water will move from its high to low concentration. So the cell is going to shrink. Water will be coming out from the cell, and as a result, the cell is going to shrink. Such a solution is known as a hypertonic solution, and if we put a cell in a hypertonic solution, it will shrink. Moving on to isotonic solution, where the water concentration is the same inside the cell as well as outside the cell. So the water that will come inside and the water that will move outside, it is going to be the same, and the cell will neither shrink nor swell up, right? And that type of a solution is known as an isotonic solution. So what are the advantages of osmosis? Where do we actually see osmosis around us? So most of the freshwater organisms and plant cells, they gain water, they acquire water through the process of osmosis. Absorption of water through the plant roots, that also takes place through osmosis. Diffusion, if we talk about, is essential for the exchange of gases and water in the cell. So it plays a major role in the exchange of water as well as gases in and out of the cell. Now, if we talk about the structure of the plasma membrane, the plasma membrane is not a rigid structure. It is quite flexible, and it is made up of lipids and proteins. And this flexible nature of the cell membrane, it allows it to move. It allows it to be a little flexible in order for the cell to engulf certain food particles, certain materials from its external environment. And this process is known as endocytosis, the process of acquiring something from the external environment to inside the cell, as you know, in the method of engulfing it, is known as endocytosis. And amoeba is an example that performs endocytosis. Amoeba acquires its food through endocytosis. Now we talk about another component of the cell, which is cell wall. Cell wall is only present in plant cells. So it is the outermost covering of plant cells, and it is present outside the plasma membrane in case of plant cells. Plasma membrane is a little flexible structure, but if we talk about cell wall is a rigid structure. And in plant, cell wall is composed of a substance known as cellulose. Cellulose is a complex substance which provides strength to the plants. It provides mechanical strength to the plants. So generally, what happens is it provides a protective layer against the structural layer, a layer which provides strength and rigidity and support to the plant cell, right? Now, if a plant cell loses water through osmosis, we just studied a case in which a cell shrinks or loses water. So what happens is the shrinkage takes place in such a way that the contents of the cell, they shrink away or they contract away from the cell wall, as you can see. right? The contents of the cell, they have detached from the cell wall. They have contracted away from the cell wall. This phenomena is known as plasmolysis. Okay, this is known as plasmolysis. And cell wall, what is the importance of cell wall in the structures that in the cells that they are present? So cell wall, not only it is present in plant cells, it is also present in certain bacteria, it is also present in fungus. And what cell wall does is, because it is such a rigid and strong structure, so if a cell is kept in a very dilute solution or a very hypotonic solution, as we discussed. So animal cell, if it is kept in a hypotonic solution, it will swell, and eventually it will burst because it doesn't have cell wall to protect it. But what happens in case of plant cells or bacterial cells or fungus, what will happen is there will be swelling of the cell, yes, and because cell wall is present, cell wall is going to apply an equal force. So even though the cell is going to swell up, it will not burst. If we keep a plant cell, a bacterial cell in a hypotonic solution, it is going to swell up, but the cell wall is going to prevent it from bursting, right? The very first organelle that we need to discuss about is the nucleus. Nucleus is one of the most important organelles present in the cell because it has the hereditary material, which is DNA, that gets transferred from one generation to another, from parent to progeny. So it is actually the place that stores this DNA in the form of chromosomes, right? So nucleus as a structure, it is covered by a double membrane. It is a double membrane bound structure, and it allows the transfer of material inside and outside of the cytoplasm, right? So outside the cytoplasm will be present, and it allows the transfer of material inside and outside the nucleus from the cytoplasm. It contains chromosomes that are structure that store or the DNA is present inside the nucleus in the form of these chromosomes. So DNA along with some proteins, they form chromosomes, and there is certain segments present on the DNA that actually are involved in generation of traits that are responsible for certain features, certain traits, certain characteristics that we have. And these segments on the DNA, they are known as genes. DNA, when it is not condensed fully, it is present in the form of chromatin material, right? Now, if we talk about the nucleus, in case of two types of organisms that we are going to discuss, prokaryotes and eukaryotes. And nucleus is a very important structure as far as reproduction is concerned, because in many organisms, division of nucleus is one of the first steps in reproduction. Very simple organisms, right? Now, talking about prokaryotes versus eukaryotes, prokaryotes are organisms that do not have a well-defined nucleus. Pro means primitive and karyon represents nucleus. So prokaryotic organisms do not have a very well-defined nucleus. By not having a well-defined nucleus, we mean that their nucleus is not covered by a nuclear membrane, right? The genetic material which is present is naked, and bacteria represent prokaryotic organisms. They do not have a well-defined nucleus. Apart from not having a well-defined nucleus, they do not have membrane-bound organelles also present inside them. So very primitive structure. And eukaryotic organisms, they have a well-defined nucleus. What do we mean by well-defined nucleus? We mean that their nucleus is covered with a nuclear membrane, right? यूकैरियोटिक सेल में दो टाइप की सेल्स के बारे में we are going to talk about, plant cells and animal cells. We'll come back to the differences that are present between the plant cell and the animal cell in some time. Now, cytoplasm represents the fluid content which is present inside the cell, inside which all the other organelles are present. They are submerged within this fluid content, and these organelles, they are enclosed by certain membranes. So cytoplasm represents the fluid content which is present inside the cell, in which all the organelles are present. So we discussed about prokaryotes and eukaryotic organisms. Prokaryotes, they do not have a well-defined nuclear region, and they do not have these membrane-bound organelles as well. Viruses do not show any characteristics of living organisms because they do not have these organelles present. Until and unless they enter some other living organism and use its cellular machinery to multiply, they are non-living in structure. So prokaryotes and eukaryotes. Prokaryotes have a very simple structure. They are small in size. They are mostly unicellular. They do not contain any organelles. They do not have a well-defined nucleus. And examples are bacteria. Eukaryotes have a complex structure. They have a well-defined nucleus. They can be unicellular or multicellular, and they have membrane-bound organelles present inside them. Example, plants, animals, etc. Now, what is common between both prokaryotes and eukaryotes is that both of them contain DNA. In prokaryotes, this DNA is naked because it is not surrounded by a membrane, but nonetheless, they do contain DNA. Both of them contain ribosomes. So ribosomes are the only organelles that are present in prokaryotes. Apart from ribosomes, they do not have any other organelles. Both of them have a cell membrane that covers them, and both of them contain cytoplasm. Now, if we move to the structure of plant cell versus animal cell, both of them are eukaryotic cells. First main difference is that the outermost layer in a plant cell is cell wall, whereas the outermost layer in the animal cell is cell membrane. Animal cells do not have cell wall, right? Plants have plastids present in them, whereas animal cells do not have plastids. Generally, we see that plant cells have a very large vacuole present, which occupies 50 to 90% of the space inside the cell. Animal cells do not generally have vacuoles, but even if they would have vacuoles, it will be very small in size. In animal cells, we see that the nucleus is centrally placed. It is present in the center. In plant cells, in most of the cases, we see that nucleus is shifted towards the periphery a little bit. It is peripherally placed because vacuole generally occupies most of the space. So nucleus gets a little shifted into the corner. Right, so these are some of the differences between plant cells and animal cells. Both being eukaryotic cells, still we can see some differences between them. So this is all for today's session. We will complete the second part of this chapter in the next session.